We are focusing on the marbled crayfish because that's an invasive crayfish. It's a special one because it appeared only in the late 90s, artificially, because it was by hobbyists in Germany that created a new species out of another one. Procambarus phallax. It's a North American species, and they breeded with it and they played with it and they generated a new species. And that escaped into the wild, and that became a big problem. Why is that such a big problem? Because it reproduces artificially, asexually, and that is an enormous advantage if you want to colonize new habitats. So you can spread really fast. You don't need a partner, so they produce eggs by themselves. There are no males involved anymore, so they can really easily spread across the environment, and that's really a painful thing for the managers. They think that they have a very detrimental effect or impact on the native communities, and especially on amphibian communities. And one of the populations or meta-populations that we are studying now is in an area that is a nature reserve where the crayfish got in, and they are now distributing very fastly across that environment. This is a very diverse nature reserve with a lot of ponds and fens, harboring a lot of rare and endangered amphibian species like great crested newt, the netterjack toad, moor frog, and so on. So, and it is believed that the crayfish will have a very detrimental impact on those amphibians. It destroys their eggs, maybe also the larvae, and we are now trying in our research project, in the case study, to study the impact of the crayfish via eDNA on the amphibian community. The crayfish, the marbled crayfish, is also believed to be a carrier of a pathogen, and that is the crayfish plague. So that's a kind of fungus that attacks the native crayfish, and it's really harmful for them. So that's also an additional problem that's coming up with this invasive crayfish. First of all, we want to, because it's difficult to monitor this species via traditional methods. It hides below the water surface in the vegetation and so on. So we want to use eDNA and implement a very robust protocol to detect it, also to quantify the populations, to quantify the populations to see what the impact is also, and quantifying also the native amphibian populations, and to measure over time what the impact is of those introducing crayfish populations in the native communities, amphibian native communities. One thing that really surprised us, and also the managers, is based on their traditional methods, they called the fight nettings, they thought that the distribution range of the species was situated in a certain area, and they put a fence in it to protect the area further on. But based on our eDNA, we saw later onwards that the crayfish already crossed that area before the fence was placed, and that it was further introducing the very intact nature reserve where the breeding ponds of many of those amphibians was. Another thing that was really surprising on the crayfish study that we are now carrying out for 2 years was the dramatic increase of the eDNA in all the ponds that were infected or were infested by the crayfish over only 2 years, indicating that the populations are really massively booming. And on the other hand, we saw in contrast a massive decrease in the concentration, the eDNA concentration of the native amphibians, which is also not a good sign we are afraid of. The eDNA is a very powerful tool to monitor hidden species below the water surface, and that you can use it for many purposes. So you can monitor the problem on itself and quantify the problem. You can also help managers to coordinate their work in the field and also to evaluate their work. So we can take samples before an eradication action, and we can take samples afterwards to see how successful it was. Was it completely destroyed, the population that they want to get eradicated, or did some individuals left? So eDNA is really helpful on each of these aspects. The key takeaway message might be that we think about not to release such invasive species in the environment after we need to try to find them and need to eradicate them. So that might be maybe the first key take-away message. We are helping now. I told about the fence that the managers dropped in the nature reserve to prevent further dispersion of the species, but there are some pioneer populations that cross that area already. So we pinpoint these populations in the landscape to bring the managers to those ponds to act to eradicate these ponds and prevent further spread in that area. So that might be one of the actions that where we help them with. The local province is involved in the whole problem, in this problem, and they are putting a lot of money in it. Also, the national government of the Netherlands is putting a lot of energy in it to handle the problem, because if it further expands, it will be a disaster for nearby nature reserves and nature in the broader sense. Compared to traditional monitoring methods, eDNA has an enormous advantage, because we can sample a large number of ponds and fens and lakes at a very short period of time. So we get a snapshot of a large area. That's an enormous advantage. We have also very low detection limits, so we can detect we did experiments with it, even not on crayfish but on other species. We can even detect one single small fish in an Olympic swimming pool. So it's really sensitive, and that's also a very good aspect on it. And it's cost effective compared to traditional methods. It's not only fast, but you can do a lot of sampling and investigate a lot of sites at once, but it also spares a lot of money in terms, if you compare it to traditional methods. It takes a lot of man-hours to put fikes in the ponds, to see where the crayfish is, to count them, to find them.